All right, do we want to get going? Okay. Well, welcome back, everybody. Today we are focusing on regulatory strategy in our second to last block of sort of hard programming as we move through the ACT Accelerator here. Really excited to have Matt Wagner here today from Proxima. For those of you that may or may not have heard, Proxima was actually started here in Houston in 2017, Matt? Was, yeah. So not that many years ago, although I think I was surprised about how many years have in fact actually passed since it was launched, and was really just trying to fill this need in the community here for a CRO partner that you can work as you're moving towards the clinic, really from the very beginning, earliest stages all the way through your trials. And Matt here came on board a couple of years ago as the head of the clinical operations and is now, I think, the VP of the CRO services. So really excited to have him here with us today to talk about a little bit about his story, but also to talk about this larger sort of regulatory path that you'll be going down. So take it away, Matt.
Awesome. Thanks, Sarah. Yeah, so I'll click through and just give a little intro to myself and my team and my background directly, but super excited to be chatting with you guys. I actually wish we were in person. I know we're virtuals, but I am excited to come to the summit that's coming up. Hopefully all you guys will be there in person and we'll get a chance to actually meet. But I'm based in Houston. I'm a Texan native here of the Houston area. But as Sarah said, we are based here in Houston in the TMC. So we are co-located in the TMC Innovation Center space is our primary office. We also have an office in J Labs. I know since the pandemic, people walking around is a little bit less than what it used to be when I first got into this space, where it was a lot more bustling, but it's getting back. So excited to see that and excited to see you guys in person. A little background on me. I started in academia myself. So I see some of you guys from Texas A&M. I'm a Texas A&M grad originally. And then started working at MD Anderson Cancer Center in radiation oncology. So I was actually working in the lab on radiation sensitizers. So we were looking at a variety of different drug products that were coming out at the time and testing them to see if they were capable of adding any sensitivity to radiation. And then I kind of moved into more clinical research, worked for a few years in orthopedics, UTMB out of Galveston, and then kind of moved back into oncology, worked for US Oncology, McKesson Specialty Health organization for a number of years, working with the docs there on their site management organization, and then in their CRO organization, of leading that CRO for a few years before I went over to another CRO called MedSource. MedSource, I was there probably about 6-7 years, and then they were recently acquired, I think, 2 years ago, but I was already here at Proxima, where I joined up with Kevin Coca here, our CEO and co-founder. He and I worked together at the US Oncology space for a number of years. So kind of joined teams again. A couple other friendly faces you might see, hopefully when we get together in person, but just love to point out our team. We got an excellent seasoned team here. Stephanie Moll has stepped up into our Director of Clinical Operations, taking that role. As Sarah said, I was previously over clinical operations, now have a broader role in the organization. And then Isabel Smith leads our regulatory team. So that's our core team that would be interacting with you guys, hopefully at some point as you move through the path. All right, so the topic that was kind of handed over that hopefully you guys are excited to hear about was kind of FDA regulatory paths and clinical trial planning. So my background, as I just kind of went through, has been really focused on the clinical development, early phase, and then all the way in through kind of late phase, all that clinical operations piece that I've been leading most of my career. But through that, I've seen a lot of different regulatory components. And then here at Proxima, we do have a full regulatory team that starts with companies just like yourselves doing regulatory strategy. So what I've done is kind of highlighted some of the key questions that I get all the time from companies just like you. So hopefully that'll fit what you guys are looking to see for some regulatory considerations and what you might need to think about, how to get into the agency, what paths, what different things might mean to you in development. and then I'll move into some clinical development planning. That's one of the funnest pieces to me, is actually taking where your product actually fits in the clinic and trying to match that up. Where are you going? So I've got a few tools that I'll kind of go over in clinical development planning. And then I've got some trial designs, some basic stuff. Might be a little bit too basic depending on where you are and what you've done in your career for different trial designs, but got some basic overviews, phase 1 trial designs, a phase 2 trial design, a little bit on endpoints as you kind of think about where you're going, what your product's actually doing, and how you might need to negotiate those endpoints with the agency. And then I'll kind of wrap it up with some just overarching study management considerations as you get into trial operations. That's a big piece. It could be a presentation all on its own. So I just got a few highlights of kind of study management considerations for you guys to think about as you're actually planning your actual studies, and what resources you may have to execute them, what type of team you might need to build, or what kind of partners you might need to partner up with. And if we have time, I'll kind of end it with a couple of kind of interesting phase. Most of them are late phase on trial failures. Just things to think about as you kind of wrap up. It's always good to learn from others' mistakes or others' things that they've ended up kind of ending their cycle on a failure note. So I'll kind of wrap up with those if we got time. If we don't have time, I'll kind of open it up. I'll try to remember to stop towards the end and just kind of open it up for some Q&A so you guys can get the most out of the time today. I don't want to be speaking this whole time, but again, in virtual environments, I know it's always a little tough to do a Q&A, but I'll try and make sure we get some time at the end. All right. Well, as we dive in, I just wanted to acknowledge the cohort here in the audience. So again, not sure, not being able to see everyone all at the same time who was able to join today, but I know within this cohort, we've got a pretty diverse group, which is fantastic. I mean, I love to see, obviously, again, Texan native, the representation of places where I've been in my career, show up on this chart right here of the cohorts. But also just wanted to recognize the diversity of products, the diversity of from targeted therapies, cell therapies, immunotherapies, you got the RNA, and then just platform groups, and then diagnostics too. So you've got a broad range audience here that I'm speaking to today. So I'll try my best to cover a few different areas. I know we're kind of heavily weighted on the therapeutic, so it's probably a little lighter on the diagnostic piece, but definitely love to follow up on the diagnostic piece too, even separately if I don't hit enough key points for that group there, to make sure I can answer any questions as we go. Those paths are a little different, as I'll point out here in a second. All right. So as we kick things off in this presentation, again, recognizing kind of where you are, where we are in oncology development today. These stats are probably even they're a little old, but just gives you an idea of how many compounds are coming through the oncology sector right now. The increase, it's still going up from the number of companies, the number of products, the amount of investment going into the oncology sector. Just continues to be a really robust area for clinical development, which is a great thing for you guys and for companies and for patients. That said, it's very crowded. So it's a crowded market in terms of trying to find where you fit in that market, how you can differentiate yourself, both to the investors as you're raising funds, but also to investigators and then patients. So as you get into the clinic, it's a crowded space in the clinic. So really finding where you fit can be difficult. The complexity of the trials continues to increase. What kinds of endpoints you're trying to track, how long you're going to follow these patients. Again, the complexity here can just be really tough. So tough space to navigate. And that's kind of, again, why we're here, why we have this accelerator to help companies like yourselves navigate the space. All right, regulatory. So just at a high level, you guys probably again know this, but just to keep it at a high level, really your product type drives where your regulatory body is going to be. So just kind of keep that in mind, know what kind of product you are. Most companies start out really knowing exactly where they are, if they're either a drug or device, but then as they get into kind of drugs, are they a drug or biologic, where they're fitting. So know your product. These are the organizations that oversee these types of products with the agency. So CDER, CBER for biologics, Center Devices, Radiologic Tells for devices. So keep in mind, these are the agency groups that you're going to be interacting with. So as you go and start looking at what types of regulatory requirements you are, make sure you're looking in the right space online, make sure you're looking at the right regulations. So regulatory oversight, I've got a couple of links here in the presentation. And Sarah, I can share this presentation when we're done as well. So the team can have some of these key links that I've included and information.
Matt, can we interrupt you as you go or not?
Sure.
I just have a quick question about those different regulatory offices. If you have a platform that uses both a new drug or delivery platform and new device, would you go to one of them and then they work together, or would you need to file for two different applications or kind of systems?
If you have a combination product, so if you're really looking to kind of understand if you have a combination product, typically you would need to file with I would typically recommend from our regulatory group, you file with your IND first, and then you would have them actually work together if it's a true combination. But a lot of times they'll give you feedback to help you understand, is it really a true combination product, or is it a supporting device? And then you will have two separate, filings that you're trying to navigate.
Okay, thank you.
Yeah, it's a good question. If you have kind of a platform technology, and if you have a supporting companion diagnostic, as some of you guys might have, so those again with a true companion diagnostic, you would actually have to have two filings, one for your drug product and one for your diagnostic product that is a companion. So those, you'll be dealing with two different filings. But if it is a true companion or is true combination, then you would have one filing where it is one product that the agency sees it as. Typically the drug agency leads, typically, in that combination. For the regulations down below, again, got some links that we can share. Key aspects, obviously, again, if you're a drug or biologic, you're going to fall under the IND regulations, investigation of drug application. If you're a device, you're going to fall under IDE regulations. So you're looking at IND Part 312, 21 CFR Part 312, IDE is 812. But always keep in mind, there's also additional regulations in addition to that. So I always want to point that out to new groups. Electronic records, electronic signatures. So Part 11, protection of human subjects, which is really informed consent, it's Part 50. Financial disclosures, 54, IRBs, 56. So those last three are really key areas that your investigators are going to follow, but as sponsors, you should be aware of those requirements as well, because it'll be your job to provide oversight of those sites that are enrolling patients on your studies. So always good to have a good complete view of the regulations, both of you as a sponsor and then also of those investigators and what their requirements are as well. That's a pretty broad set of rules, but during your clinical development planning and timeline, you got a lot of time where you can, again, get up to speed and find partners who can help you navigate all of these different regulations. All right, I have the next couple of slides on opportunities to engage with the agency. It's another key question that I get a lot is, at what points can I actually talk with the agency? When can I get some good feedback? You guys are meeting here in the accelerator, getting good feedback. You're meeting with investigators and potential key opinion leaders, getting some feedback. A lot of different groups you're connecting with, but when can I actually connect with the agency? So there's a basic outline. Again, it's not probably the end all be all, but it's a basic outline of key meeting points with the agency. So if you're a drug biologic, you'd have the IND pathway. Then you can have a pre-IND meeting with the agency. Down below is a little bit of information of what you need to submit with that pre-IND request and then your other meeting requests. But that's really your first touch point with the agency. Within that, you're usually going to have a list of questions that you're going to ask. So you're asking for that meeting. You can ask for it to be in person or just a written response. Most recently, the agency has been providing written responses in lieu of meetings, even if you did ask. So it kind of depends on the division that you're working with, but I always recommend an in-person or teleconference type of meeting. If possible, you get good feedback in those meetings where you can actually have dialogue. But your first chance is typically a pre-IND meeting. So actually filing your list of questions, submitting that to the agency, getting feedback on your product, on your preclinical data or preclinical plans, and typically on your study design as well. So those are some key areas where you can ask a list of questions and get that feedback to the agency and begin your interactions. Of course, when you submit your application, you're going to get feedback there as well. The agency will reply back, let you know what kinds of gaps you are, but hopefully you have submitted a pre-IND meeting and you won't have any surprises when you actually submit your IND and you're ready to actually run your study. Other opportunities, you do have an opportunity for end of phase 1 meeting requests. Take advantage of each of these opportunities, get an end of phase 2 meeting request, and then you can obviously do a pre-marketing submission as well.
Matt.
Yeah.
This is Enrique. I have a question for you. For the previous slide, for end of phase 1 and end of phase 2, is there any additional approvals that the company must obtain from FDA? Are there specific authorizations like to move into phase 2 if phase 1 went well and so on?
So for each of your studies, you're going to submit those protocols to your IND. So with each of those, you don't necessarily have to have the meeting request where you're actually having feedback. But again, it's a good opportunity to get feedback, especially as you're planning your studies. So at your end of phase 1 meeting, it's a good idea to submit some of your data, and then you can again ask key questions to the agency directly without having to be fully prepared to submit your next protocol. But you can ask, let's say you're going from a single agent into a combination. You may want to understand, do I have the preclinical information to justify that combination in human? You may want to push the boundaries and go straight in without preclinical animal data and combination. So that's an opportunity to ask questions, but you can't just submit your protocol. Enrique, if you feel pretty confident, you're like, I'm going from a single agent phase 1 to a single agent phase 2. Your dose, it's pretty straightforward. You can just submit your protocol under your IND application. And then if they don't reject that protocol within 30 days or give you feedback, then you should be good to go.
Thank you, Matt.
And I didn't put a lot of the timelines, but there are key timelines when you do submissions. Each of these requests, whether it's a meeting request or an IND application itself, there's timelines that the agencies will have in writing that are in the regulations that they're supposed to stick to. So they're supposed to respond within that 30-day window when you submit your IND application with feedback and let you know. Typically, it's very cordial. Hey, they're like, hey, you're missing this. I don't see this. What is that? Where's this information? They're trying to review it within that tight window for them to give you feedback. Or they might say, hey, you're missing a complete section. We're going to place it on a clinical hold until you provide the additional information that you can provide. All right, since I know we did have a diagnostic company here, I did want to mention this for medical devices. It's very similar. The key meeting that I'd say for medical devices, of which there's several different types of meetings, so I didn't list all the different types, but they basically have a Q submission process, and has a list of several different types of meetings. Key meeting that I recommend is a pre-submission, a pre-sub meeting. It's basically very similar to your pre-IND meeting or your other time points, as opposed to different phases with devices. You don't have those same phases organization. This pre-submission can be done at any point in time, but it's structured very similarly. You're basically going to propose an agenda, you're going to ask questions and get feedback from the agency directly. So you can do that. You can ask, like, do I need to file an IDE or not, because again, there's different rules around being significant risk or non-significant risk with devices. We'll go into all the details of that today, but again, I can follow up on that with that particular group. I want to mention pre-subs is a key meeting request point to get feedback from the agency if you have a device or if you're partnering with a company doing a companion diagnostic. So you'll want to take advantage of those meetings. You can ask questions such as, are you really truly going to be needing a companion diagnostic with your product? So you can do that at any phase. It may not be necessary to do it in phase 1 when you're doing your early phase study with a drug, but then later you're starting to realize, hey, this is going to be a biomarker where it looks like it might be a companion diagnostic. Now's a good time to do a pre-submission meeting with that companion diagnostic and get that kind of feedback. The structure is relatively similar of the meeting request, file a meeting request, list of questions, whole briefing package goes with it. But it's a good chance to get feedback at any point in your development cycle with the product. All right, this one is always a fun one with questions around what are all of the different opportunities for regulatory pathways and expediting your program, right? So these kinds of designations are ones that get you excited, they get investors excited. So you're trying to understand what they are, what they mean to you, and what do you need to get them. So here is a quick kind of chart. This actually comes from the guidance. I've simplified it a little bit here, so it's not quite as wordy as the guidance itself. And there's definitely, again, more criteria to be aware of, but this is the key aspects that I thought kind of stuck out. What is this and what is it to you and your product? All right, so fast track. It is a designation for that product and indication. So what do you need to get fast track? It's going to treat serious conditions, and essentially with fast track designation, it can be non-clinical or clinical data. So in this case, you can file it early. You can file it with your IND. You should probably, again, file it before you get all the way through your programs, because again, it's a designation that's going to have a few different advantages to help speed you through the process. Essentially, get you a little more attention from the agency, something to do press releases on. Then breakthrough designation, breakthrough therapy is another designation you can get. This one does require preliminary clinical evidence. So that's what's a little bit different about that breakthrough. So you're actually saying this is a potential breakthrough for this indication based on some of the clinical data that's come out with your product. Again, you can submit that application early with your IND. Some products I've seen have clinical data outside the States before they have it in the States. So they may be able to submit it right away with their IND. But typically, they're going to submit it probably after their phase 1, once they have some first-in-human clinical data to go with it. All right, an accelerated approval. This actually is a pathway. So this is an actual approval pathway for expediting your product to the market. So requirements are similar. Again, these are all around being serious conditions. So it has to treat a serious condition, and then it has to provide a meaningful advantage over the available therapy. That is a bit subjective. Here is kind of what defines meaningful advantage. Essentially, that is a discussion you would need to have with the agency during your clinical development life cycle. So typically, once you have probably already said to the agency by having a fast track or having breakthrough with some clinical evidence, now you're working with the agency to say, is this, can I get approval for accelerated pathway? You're going to end up negotiating on your endpoints. So you're looking at a surrogate endpoint that can then be followed up by an actual traditional endpoint. So for this type of pathway, you're really going to be negotiating that probably in your phase 2, phase 3 planning to know what the agency would agree to. So I've got a slide a little later on that actually goes through some of those endpoints and which ones they consider more traditional endpoints and which ones they consider surrogate endpoints. But just as an example, like your traditional endpoint might be overall survival. That may take, years to actually get to, but the agency might accept here a surrogate endpoint that they actually, again, would approve, again, based on the fact that it's serious and that it provides a meaningful advantage based on the clinical evidence that comes out with that surrogate.
Hi, Matt. We have a question from Stephen Lee. He says, "Hi, Matt. Do all these designations also apply for diagnostics lab developed tests?"
So these are related to drugs in this pathway. So drugs and biologics. So anything that's going to be in that IND pathway. For devices and diagnostics, there is a couple of different designations. I don't have a list of all of them here, but the key ones for diagnostics, they do have a breakthrough device designation, very similar here to fast track and breakthrough, where you can actually apply for that. It has to be, again, meeting some seriousness criteria to get that breakthrough designation. So it'll have to kind of meet some very similar, but it's a little bit different for devices. But it does have the breakthrough device designation. If you don't qualify for breakthrough, there is a new program called the STEP program. It's kind of a step down from breakthrough that you can also apply. It's a little bit less on the criteria of the seriousness. But those are the two kind of main ones that I see for devices, is breakthrough device designation and then part of the STEP designation. All right, the last one here, another designation is a priority review designation. Again, this one, again, treating serious, and it provides improvement of safety effectiveness. So again, you're really getting this later in your data set. So you're really going to submit this with your actual marketing application to get that priority review request when you send it in for the agency. Or you can get priority review if you have, again, a pediatric designation that you're adding to your label at a later point, or unless you're going straight into that pediatric indication. So that's the priority review, but that's going to be right towards the end when you're actually submitting your marketing application. All right, so the other one that I see here, and I see a few different the groups that might actually fit into some of these indications. That's why I kind of wanted to go over them to you guys today, but orphan designation. This one's a little bit different than the seriousness criteria that we just went over. So all the other designations are based on really that unmet clinical need, the seriousness of the disease. So if it's a life-threatening or fatal disease, of which cancer meets that requirement, that's where all of those other programs come in. But orphan designation is actually meant specific to rare diseases. So it's another designation you can get that's very specific to what are you actually going after and how rare is it. So this is the definition here at the top. Rare disease, a disease affects less than 200,000 people in the US. So that's the US's definition, the FDA's definition. There is other ex-US definitions that are very similar, but I don't think they use that exact number. So just kind of keep that in mind if you're trying to compare US and say Europe or other countries in Europe. Really, the key considerations here is that, again, you're getting a designation per disease state for your product. So if your product goes after a rare type of cancer or 5 different rare types of cancers, you can get an orphan designation for each of those actual disease states. So you can file it multiple times. So I've seen companies actually plan that out in their development cycle or in their funding cycle to actually have something press worthy to submit at different points for that company to have awareness around their company and their product. Other component, just key considerations, it requires some scientific rationale, but does not necessarily require clinical data. Clinical data will definitely help, but it doesn't require it. So you have to have build basically your case for why this product will actually be useful in that indication. So you can get orphan designation based on your preclinical data set.
Matt,
Yes.
I have a question. For that designation, you indicate less than 200,000 subjects in USA. Is that the new incidence for that disorder or the total population suffer from that disorder? Because some of them linger for years.
Yeah. This, I believe it's the total amount of patients that it affects. So this is what makes it, again, a tough one, but you have to have that data. But you can go find that online from different data sources and actually pull that of how many patients are in the US with that indication.
Thank you.
Noting targeted therapies, the FDA typically does not recognize those subpopulations, or I will say it's difficult for them to agree to without additional scientific rationale. So if you have a targeted therapy and you're looking for that scientific rationale, you would really have to work hard to prove that your product would not work in the broader population. So if you're looking at, say, non-small cell lung cancer, working with a client in that space, if you have a targeted biomarker that you're going after, you would really have to show that, again, your product doesn't work in the other areas of non-small cell lung cancer versus just your targeted population for them to agree to that orphan subset. So it's pretty difficult to get that, to build up that type of rationale until you're really late in your program. And then you have to evaluate, does it really add any benefit to file an orphan designation until you're getting ready to submit for other types of things like accelerated approval or priority review already at your marketing application. The key incentives, it does have tax credits for your clinical testing. There is a big credit there for the waiver of the PDUFA fee. And then it does have some exclusivity advantages as well. So really a lot of key advantages you consider when you're doing orphan. A lot of companies like ours will help you do a little bit of that background research, evaluate, is it fairly quickly, is it going to qualify for orphan or not, based on the latest epidemiology data that you can get out there publicly. It's a pretty straightforward application, I would say, just because of the requirements are based on scientific rationale rather than hard clinical data. All right, any other key questions on any of that? I know I'm kind of marching along, you guys asking a few questions. Before I go on to the next section of just kind of putting together your plan. Any other questions?
Matt, Enrique here. Can you have multiple designations for one indication?
Yeah, so if you're looking for multiple designations across each one of these different designations, you can get at different points in your pathway. So there's no rule that says if you have one, you can't get the other. And really, you're looking at that particular indication and condition. So you're looking at that disease state there, you're treating that condition. So you can, again, get it an orphan designation for ovarian cancer. I believe that's orphan, I can't remember off the top of my head, but if you're getting it as an example in one cancer type, you can also get fast track in that and breakthrough in that cancer type. And then you can also then apply for other cancer types. So a lot of the oncology indications will qualify for these that you see right here, because again, they're treating serious conditions and unmet kind of clinical needs and spaces. But the orphan is where it gets into more of the rare subtypes or rare types of cancer. But you can apply for as many as you feel like you qualify for.
Matt, can I ask a question?
Absolutely.
I don't know if you mentioned this, I may have missed it. So this orphan designation could be used to define a subset of certain tumor based on biomarker, or it has to be based on this already existing group of cancer.
It's typically based on that existing group of cancer. So that a cancer set there is a part in the application where you can apply for an orphan subset. So if you do have data, but you would really have to build scientific rationale that their product will not work in the broader group.
I guess, sure. I guess that's related to the number. So if it's a subset, then it's possible, certain cancer can have less than 200,000. Right? But if it's a whole population of certain cancer, then only very few cancer type can meet this number. But I guess I get your point. I have a good case. Right, thanks.
Yeah, and the more actually, the better we do with cancer therapies these days, with patients actually surviving longer, the less there actually are of those that are considered that fit into that orphan category as we go. I can't remember the indication off the top of my head, but I was working with a client recently. We were evaluating orphan designations, and one of them was just on that cusp. The newest data came out, and then it became above 200,000. So it was, 215,000, as opposed to a previous product a year before had recently gotten orphan designation in that indication, but now they could not.
Gotcha. Thank you.
Can I ask a question here?
Sure.
So, I mean, clinical trial, we have a basket trial and umbrella trial, right? So, which means it's whether it's depends on the disease classification or it depends on the target classification. So, if a target specific protein pathways, but it can be used to treat multiple diseases like prostate cancer or breast cancer. So, this should be a single application, or actually, single orphan disease designation or multiple.
So the orphan designation is per actual disease type. So as you just kind of mentioned, your protocol may be designed as a basket or umbrella, again, depending on how you want to set up your protocol. But for the designation, it is per disease type. So you can apply for that orphan designation in whichever actually meets the definition of orphan. So like breast cancer, typically, again, unless you can somehow prove the subset is orphan, doesn't qualify as orphan, but that can still be a cancer type in your protocol. So these designations don't have anything to do with your actual trial design. They just have additional benefits where when you apply for that actual disease type.
So that means that there has to be 2 applications in order to treat 2 diseases.
You would have an application. So you may file. I guess let's just clarify which type of application that we're talking about. So if we're talking about orphan designation, if you have your IND, your initial IND, you have one IND that you submitted, and then you can have multiple protocols under that IND initially as you're doing your planning. And then as you do these different designations, you're gonna have a designation per, again, disease state. So these aren't going to be directly tied to your protocols. So right, as the example you have again right now, so we have orphan designation where we filed 3 orphan designations for a particular client, but in their protocol, they're planning right now, they're going after, I think, about 8 different indications in their protocol.
Okay, good, thank you.
Awesome, great questions. All right, we'll move into just kind of highlights of clinical development planning and a little bit of tips on data collection as we kind of go in there, and then we'll go into some trial designs. All right, for clinical development planning, I always like to start discussion here with what kind of got stolen over from AZ, AstraZeneca's framework that they published on a few years ago. They called it the 5 R's of the development process. So they're looking for a right target, the right tissue, the right safety profile, the right patients, and of course, right commercial potential. So this is a good way to break it down. And then the next couple of slides, I've got some actual tangible examples, how we've kind of helped clients actually organize their data to help look at what they're actually collecting both preclinically and then clinically. So starting at that target, looking at a strong link between what your product's doing, what it's going after, and the disease state. So you want to have differentiated efficacy on what your product actually can do. The right tissue, really looking, is that tissue available? So a lot of different companies now are looking, again, what's the product delivery that you need to get into that tissue? Is it a direct intratumoral injection that might be the best case? So what tumor types have the availability of tissue? How easy is it to access? And how is your product actually working? What's the PK/PD profile on that particular tissue? Safety, obviously clear safety margins, making sure you have the right risk-benefit ratio for your product. A lot of targeted therapies these days are getting safer and safer, so that one's less of an issue, I think, but always have to keep that in mind, the right safety profile for your product, and then where you're going after the right patients with that safety profile. Patients, again, looking at not just does that patient have your marker, but is there actually a correlation between responsiveness and your marker? So looking at finding the right patients and the right patient profile. And of course, right commercial potential. I won't spend as much time on that, but that's kind of obvious. Got to have the right commercial potential for there to be interest in that product. All right, a tangible tool here that we've kind of put together. This is an example of one that I've used to actually help with an indication prioritization exercise for clients. And it really helps to organize all the different data that they've collected, whether it's coming first out of the lab, first time you're forming your company, and then continuing that preclinical data, continuing all the research you're doing in the lab, and then kind of continuing to collect what kind of information as you go into the clinic. So really trying to break it down into chunks. Where is the science taking you? You guys are all probably scientists here on this call, so you guys know your product really well. So this tool is just a way to organize it, and a way to kind of say, how much data do you have in each of these kind of areas here in the front end? And again, this is a snapshot of it, so this isn't kind of the end all be all. This is just an example today for like how I've seen clients organize their science. What are the different tumor types that you have data on? What are the different areas where you may have biomarkers present? And there you might have columns with 3 or 4 or 5 different biomarkers that you're collecting and organizing this data. Again, this is just a snapshot here. Then you may have correlation to outcomes with that biomarker, as then you've run different models on there, whether it's in vitro or in vivo models. So where is the science taking you? Where's your best shot at that science overall? Then with that, you can kind of pair that up to what are the options here from regulatory paths that we just went over? Is there any advantages in different tumor types? Is this one orphan? Does this one qualify for that breakthrough therapy? What are the different opportunities that you might have to gain advantage by, again, taking that particular product into the clinic? And then looking at your clinical aspects as well. Is there any issues with tumor accessibility? You know, is it highly accessible? Is it really low? Is it really difficult to get to? Is it really difficult to get your product to that particular area to actually treat the disease? Looking at the clinical landscape, what's that incidence, prevalence, number of deaths that you're seeing in there? So looking at that kind of data at a snapshot, this really can kind of help essentially boil the ocean down into something tangible that gives you a quick view or your team a quick view to look at where is the science taking you, where are your shots on goal in the clinic, what type of advantages regulatory you have. And then towards the end, as you get into actual clinical trial planning, you can add in other components like the logistical considerations or operational considerations, like how many competing trials are there in that space? Is this ridiculously crowded at this point in time that you're entering the clinic? So you can't really underestimate that. So I always want to point that again, I'm a trial operations guy in my career for the most part. So keeping that in mind, I've seen some excellent science get stalled because they went after an area where someone else was already doing 3 phase 3 trials. So they really just couldn't get anyone to sign up for the study until that was done. So your timing you want to kind of keep that in mind as you get into the clinic there as well. So just things that you can kind of keep in mind and really help paint that picture and help organize where you're going. You might have science that attacks multiple areas. Hopefully it looks like most of you do have different opportunities for different tumor sets, and this will help organize that. All right, and then to kind of pair that up, again, another just tangible example that I've used, kind of deleted some of the data off to the right, just to make it easier to view on the slide. But as you then start to get into clinical planning, this is another step you could take to help you understand the landscape of your tumor type. Now that you've kind of narrowed it down into a few different tumor types, I really recommend obviously understanding the landscape in the clinic. Where would your product fit? Where are they using other products that you may want to go into combination with standard of care? So that way you can understand, okay, what are the preclinical studies that I might need to be doing now to understand drug-drug interaction, to understand if there's any advantages for different tumor types over others when I start doing it in combination. So as you have some preclinical data in non-small cell lung cancer, learning what are the other drugs that are in non-small lung cancer and where they're actually used. Are they use adjuvant therapy, they use first line, second line, third line, or they use maintenance, and then understanding what types of products those are. So that this kind of takes it a step further, allows you to do a little bit deeper planning and understanding what types of combinations you might need to consider. Or if you've got data that can take you into a specific area without going into combination, that's another consideration you can kind of look for. Again, you may have data that says it doesn't matter the PD-L1 status with your particular product. You've got good data regardless. So you may want to go against the grain there and go into one of these other lines of therapy that kind of dodges being in combination with a checkpoint inhibitor in lung cancer as an example. So really understanding kind of where you might fit. This is a great conversation piece too when you're actually talking with KOLs. It shows that you know the space. If you're talking with a lung cancer KOL, now you can actually have an educated conversation because you know exactly where these other therapies are being used. You can get their feedback where they might see your product, where they see an unmet need. So really getting that feedback and then kind of figuring out where it fits. So this is a good deep dive to understand that, especially as you get into cancer where there are other products approved that are targeted. So the subpopulations are starting to split as you get in. It's no longer just non-small cell lung cancer. It's where those drivers actually happening, what other products are actually approved there with those different subtypes. Breast cancer is kind of way above the curve. It's been doing it for tons of years here, but now other cancer types are starting to see those kind of same subpopulation splits. And so trying to understand where those products are is super helpful.
But we have a question from Johanna Webb in the chat. Does the FDA view the subtypes of breast cancer as individual disease states, or would we have to prove that we are not effective in any other subtype? Johanna, jump in if you need to clarify.
I think she tried to say is if for the breast cancer, there are different subtypes. For each subtypes, we'll consider based on the population, you can define as orphan drug or not orphan drug, or you have to, we talk about subset of the cancer. Yeah, for breast cancer, there are so many different subtypes, and certain subtype, the population certainly be below 200,000. So the question is, can we use a subtype to apply for orphan designation, or we have to prove our drug only applicable to this subset?
So with the orphan designation, you do have to have rationale that scientific rationale that it would not work in the other subset. So if you want to apply, you can apply. There's nothing against applying for that orphan designation, but in order to achieve it, you really have to have that scientific rationale. So you have to have some, what I would say, some data. You don't, again, it's not a requirement to have clinical data. You could have preclinical data that shows, again, your product will not work in the broader population and only would work in the subtype population. But you do have to build that scientific rationale so that it's fairly difficult to get that subtype orphan designation.
Okay, thank you. Thank you. Answer.
Matt, this 200,000 is the whole surviving population. So if you see that on any of these breast cancer subtypes, they have more than 200,000. Per year, there may be low, but a lot of people are still surviving the TNBC. If you count all of them, it won't qualify any of these designations.
Yeah, that's true. So I might have to clarify. I'll have to go back and check and see the clarification if it's the entire population or just the new incidence rate. So I'll follow up on that action item on my end. All right. So I know, like, recent, again, one of our recent applications tried to set that case for a subpopulation, and they denied it. They just said there wasn't a strong enough scientific rationale to say that the product wouldn't work in the broader population. So you can probably continue to build that case with more data, whether it's preclinical or clinical. But I know the agency looked at that as a pretty stiff regulation to really prove that it wouldn't at least have some strong, very strong scientific rationale that it wouldn't work in the broader population as a product. I think it's very difficult because you're looking at others that are kind of +1, +2, +3. You haven't really always defined what those categories are if it's a newer marker. So whether it works or doesn't work, there's that gray area. So I think that's really, at least in my opinion, why the FDA is kind of on that, drawing that line in the sand, is because you really can't then decide where you do on that scale of positivity of the marker or percent of that marker availability. There's just not enough data typically to paint that case yet. All right, key tips of just clinical development planning and your strategy. Again, starting kind of with the end in mind, know where you're going, know how to get there. So a lot of this information that we just kind of went over in your clinical development plan, organizing your data, knowing different opportunities for pathways, know kind of where you're trying to get to, and that'll help you design the best studies to get there. So really should not start a study without knowing what your next study is going to be. You should really be looking always a step ahead or 2 steps ahead, kind of knowing where your end game is going to go. So keep that in mind when you're doing your planning and what types of data you're going to need to get there. Even when you're getting into that first in human study, it's a huge milestone, but if you already know you're going to need to be in combination, you should be planning for what that combination going to be looks like, what type of preclinical data you have to support that combination as well. Again, I've seen studies go into the agency with a phase 1-2 design, and they kind of went, or phase 1A and 1B, where they went from single agent combination. The combination didn't have any preclinical data, so the agency kicked that back and said, I'll let you do the single agent, but they had to amend their protocol and go back and get some animal data before amending the protocol. So just some examples of why it's important to know that, because it'll save you time and headache along the way too. All right. Just some general trial design considerations, really helping pair up your design with how you're going to analyze your data. So the next kind of points are on your protocol and data collection. You really should have these kinds of things discussed and finalized before you start your study, at least to the best extent you can possible. Clearly your design, how you're defining definitions such as progression, how you're going to analyze the data. You'll typically want to have your statistical analysis plan there shortly after your protocol. You Some do that right away. I always recommend to do that cyclical analysis plan up front with your protocol, so you can really understand again how you're going to analyze that data, how you're going to analyze any missing data. Patients are going to be followed for a long time. Clearly, patients are people too. They miss time points. What are you doing with that? How is that going to be censored? All that should be defined in your statistical analysis plan. So it's good to negotiate those kinds of points with the FDA early on, so there's no surprises later. And it's good to understand what type of data you need to collect. Got some points on that in just a second. And then understanding if you have need an independent endpoint review committee. So if you do need to have that independent review, such as independent imaging review, then that's always good, again, to negotiate that up front. Are you going to need that as a requirement already in your phase 3? So what are you kind of planning for? All right, now where everything kind of meets the road, where kind of rubber meets the road, is the protocol. Essentially, your protocol, you've got to treat it kind of as your study Bible. It's your end all be all on your study. The more time you have on your protocol, the better quality that you put into your protocol, the clearer you are with the actual operational details as well as the design. The less headaches you're going to have on the back end. So really making sure it's very clear actually what you're doing, how you're doing it, when you're doing it, who's doing it. So if there's things that are clearly delineated for patient visits, there's clearly delineated for stopping rules within your protocol. There's operational detail that goes along with it. It's kind of the key thing that I usually see missing in protocols is additional operational detail. Oftentimes the schema is very clear for patient flow and the schema of the protocol. But then when you get into the operational detail on exactly how those other things are going to fit, isn't always outlined in detail. Remember, the investigators obviously are the head, but their coordinators are the ones actually reading this protocol and have to understand it as well. So they have to know how to execute the details of the protocol. And some things you may have in supplemental components. So it's good to understand whether you're going to put certain details in your protocol. If you're going to have a supplemental operations manual, that is okay. So don't think that you got to put every single thing in the world in your protocol, but you need to be clear that, hey, this detail is going to be followed up in a study operations manual or a tissue collection manual, or you're going to have a pharmacy manual. You're going to have additional information that's going to support that protocol as well with those types of details. All of those types of details so that you can actually execute have to be extremely clear for the sites and the staff. And it doesn't matter how complex your protocol is or your treatment or therapy. If a site can get it wrong, sometimes they'll get it wrong. Again, there are people as well. To our company that's doing diagnostics here as well, I've seen just as many mistakes, if not more, on the simplest diagnostic study that you can imagine, where it was all they needed to do was to have a blood draw and then send it off to wherever that lab was and find out via the tissue if the patient had cancer or not. It still had mistakes of the sites because the protocol wasn't clear of some additional operational details and key time points where they needed to collect the time point information of exactly what time points those things were drawn for that blood draw. So keep in mind, it's not just the simplicity of your product, it's just the details of how you're actually going to execute the study here. It's so important. Circulating your protocol with key people and multiple people really is super important as well. You guys probably know that, of course, of submitting it to different oncologists, different investigators, KOLs who you're going to work with. But definitely make sure to submit your protocols to other partners or vendors that you're working with as well. Even if you don't think that it's that important, they can give you just independent review and an independent advice to say that, like, if they read something and they didn't understand it, they're going to give you that feedback as well. So We've done that with whether it's us as a CRO, done that with other kind of labs, even groups as a vendor. If you have a central lab or central core lab, like an imaging group, they're reading it with a different mindset and different set of eyes. And so they'll give you some key feedback on where some clarity might need to be added into your protocol. So don't be afraid to share your protocol. I know everyone's always afraid of things getting out or something like that. Make sure you got confidentiality in place, of course, with whoever you're working with. But don't be afraid to share it with the people you're actually going to be helping you execute on the protocol. All right, I won't spend a ton of time here. We'll start kind of moving through this a little bit faster as we go, but just here's some points on data collection. Really, the key thing here at the top that the FDA kind of pointed out in one of their guidances, data collected should be sufficient to evaluate safety and effectiveness of the treatment, but doesn't need to include other data. It needs to just be, again, kind of fit for purpose, if you will. But again, you're going to need certain elements of data depending on how you're going to do your analysis and what different types of analysis and breakdowns. So clearly you need some basic information at the front too. So here's some key points. There's a link to the guidance I'll provide as well where a lot of this came from. The great thing about oncology is there are a lot of cancer specific guidances out there and available too from the agency. All right, other data collection considerations as you move forward with your protocol, think about how it's going to be collected as well. So think about what types of case report forms you're going to need, how you're going to capture that data. If you have additional data points that's outside of a traditional case report form like a central lab, how's that data going to come together? So think about data flow or talk with your particular provider who's going to be providing that service for you, how that would flow. You may be collecting quality of life information, so patient reported outcomes. You may need a diary of that patient, whether it's symptoms or treatment diaries. And of course, going back to regulatory, you always have to make sure it's a compliant system. So always ask those questions. There's a lot of systems out there these days, but just make sure, again, they've been validated appropriately before you use them. All right, now I'll hit on just a few trial designs. I know it sounds like you guys are already thinking ahead to different trial designs. Love to follow up on trial designs after this as well. If there's any kind of key questions or thoughts you want to run by when you're designing your studies, but just I'll hit the highlights. Sounds like most of you guys are in that kind of early preclinical stage, getting ready to move into the clinic. This is just a basic snapshot of the drug development life cycle. As most of you guys probably know, you're either in drug discovery, preclinical, and then you move into clinic. Typical phase 1, 2, phase 1, 2, 3 studies, and then approval post-market phase 4. For diagnostics here, typically, again, you don't use phases when you're working with medical devices. Usually work with either pilot feasibility studies and then into your pivotal, your clinical validation study that you'd be working with if you have a medical device like a diagnostic there. So different nomenclature, but in principle, you're kind of working with some similar principles. You're either collecting early data that might go into evaluating your product or even refining your product, and then you're going into a confirmatory study at the end. All right, basics, layouts of the phases. Your phase 1, your primary really is to determine your dose and your safety. It's typically your phase 1 goal. Yes, you're trying to get early efficacy as well, of course, in this, and you always should, but your goal from the agency perspective is safety and establishing the dose that you're going to take into your phase 2, which then you're going to take that recommended phase 2 dose and make sure it actually has activity within that cancer type. And then phase 3s are your confirmatory studies, again, making sure that it's actually effective to get approval there as well. So. All right, phase 1. So as you get into the clinic, what's your kind of first goals? What's your first endpoints? typically, again, these are examples. So again, not every study has to follow this exact. So if you see something that's different, there are different study designs, there are different endpoints, but for the most part, you're looking at your primary goal of understanding what is the appropriate dose of your drug or biologic to take into your phase 2. So typically you're looking at endpoints such as your MTD, maximum tolerated dose, or your recommended phase 2 dose. Again, if you see toxicity, that's where you would hit a maximum tolerated dose. If you don't see toxicity, you're probably looking for, again, some activity or a max amount that you can deliver, which would end up being your recommended phase 2 dose. So there's some parameters you can set in effect for your recommended phase 2 dose. You're going to be evaluating any dose limiting toxicity. So there's specific criteria around those adverse events and safety events that are going to be dose limiting. So understanding what those are is truly going to drive your safety profile. Then you're also going to evaluate basic pharmacokinetics, how your drug is actually working within the body. Again, you're also collecting evidence of response, but it's typically not your primary goal in your phase 1 study. So those goals you're typically going to pair up with your endpoints, as I mentioned, kind of DLTs and MTD, or your recommended phase 2 dose. All right, your general population, usually we see this pretty broad, but again, if you're in a specific targeted therapy, you may go into certain subtypes. It may not be all solid tumors as an example. It might be very specific subsets that you're looking at. You're typically looking at relapse or refractory patients who've exhausted their other therapies in your phase 1. So you're looking at essentially the healthiest patients you can get who have exhausted their options and are looking potentially for a trial here. Excuse me. Number of patients typically small, but again, depending on how you do your subtypes and how you design your study, it can actually get quite large over time, especially if you end up with some different designs. We'll kind of go over here in just a second. So some basic criteria, again, there's different ways to look at this, but this is kind of, again, just the basics here. General design, typically it's open label, not randomized. Dose escalation design, you're escalating the dose. Your typical starting dose, 1/10 of the lethal dose, and the most sensitive species tested, so that's at which 10% of the animals actually died. So it's unlikely to cause toxicity. So it's typically your starting dose. If you're looking at pediatrics typically start at 80% of the adult MTD. You're typically increasing that dose gradually, and this is the most common scheme, modified Fibonacci scale. So just as an example, so I know there's some other designs out there, but this is kind of a typical increase in dose. So here's the standard kind of 3x3 design. A statistician colleague and I kind of laugh about this design. It's not really statistically driven. And so why this design's been around so long, it's kind of hard to say, but it is still in use. There are a lot of companies, so if you submit this design to the agency, it's still a very common design. A standard 3x3 design. You're essentially enrolling 3 patients at a time at a dose level. You're evaluating for at least one kind of cycle of therapy, depending on how you define that, depending on what your product is. Maybe for a period of time, if you don't have it defined, if it's just continual, say you continual oral dosing or whatever it may be, at least like a 30-day cutoff to evaluate toxicity. If you have no toxicities, you go on to the next dose. No dose limiting toxicities. If you have dose limiting toxicity, then you are going to add 3 more patients at that same level and see what the outcome is out of 6. If you have 2 or 3 that have a dose limiting toxicity, you're typically going to stop. So it's your basic 3x3 standard design here. I'll need to probably spend too much time on it, but this is kind of your basics. It actually still is in use today.
How many doses are you going to be testing until you reach the maximum tolerated
It depends on the product itself. In your case, so typically, as you kind of look at your starting dose, this is how you would typically increase your dose levels at the bottom. So you'd have your basic lowest dose. So it's typically kind of that LD10 there, where you'd start from if you're using, again, this scale. I've seen it where companies will plan for around 5 doses where they think their product may have a toxicity profile when they reach it, but they really want to look at their in vivo data and see what kind of toxicity issues they've had. If they have a really clean profile where they could increase a significant amount without, they're also going to really need to look at biological activity. So they're going to track the PK. Where do they feel like they're actually hitting a sustainable limit of product at a PK level then as well, and evaluate it in addition to toxicity. But most of the clients I work with have at least 3 when they start with. Some have in that kind of 3 to 5 range that they plan to test and evaluate. And then of course you can amend your protocol as well. So based on that data that comes out, if you have, again, no toxicities, you're not going to keep escalating for forever. You're going to make a decision again based on some of the other activity of the product in the body. All right, so other design I just going to put up as an example here, because it's one of the other newer designs, but it's actually been around probably longer than I actually thought. There's a great website where you can actually go and play around with some of the designs. So if you go to trialdesign.org, we got the link down there. You can actually look at different trial designs. You can actually put in different parameters of your study, and it'll actually help you come up with a nice little table for your protocol. So the table down here, again, just an example, but it actually help you come up with what those parameters are for your design. It's a great tool. Had a statistician show me this a couple years ago, where you can actually go in and see some of that information, play with that, and optimize your design. This phase 1 design, this Bowen design right here, it's kind of similar into a 3x3, but it takes it to another level where you're actually looking at it statistically here and looking at the number of DLTs as you go. And I've seen this study run patient by patient, where they enroll patients sequentially. And I've seen this study design actually run still in cohorts of 3. So where they can actually treat a cohort of patients at 3, but then they're looking at that DLT rate. And then as they, again, increase their dose to the next cohort level, essentially has a decision tree that you're following. Now, do I go up? Do I go down? Do I add more patients? Do I go up, add down, or add more patients? So you may end up going up 2 levels, and then you may go back down 2 levels. So you're actually following that. And what the statisticians I've spoke to actually helps you get to a tighter dose that you feel a lot more confident in taking into phase 2. So there's definitely some advantages here statistically in this design. So the statisticians I've talked to, it's kind of one of their more favorite designs I've used because it's also easier to follow. It's not the only design that's out there, but just wanted to point it out because also from a ClinOps perspective, it's still fairly easy to follow like your traditional 3x3. There's obviously some other modified designs that are out there and available, and that the agency will accept different designs. These aren't the only designs. Keep in mind, again, these are just examples from experience here to look at and consider. And then you can kind of go playing around with a few different designs at that website. So it's a fantastic tool that's publicly available. I think some guys out of MD Anderson or one of the academic centers actually helped build that website. All right, as I mentioned, there's some alternative designs. Won't spend too much time on them, but just to kind of keep in mind, again, there's other designs that are out there. What I've shown isn't the only design out there. There's definitely different modifications on here, especially in different adaptive designs here to maximize patients and maximize what patients are actually getting. A lot of the designs, I will say, in the early phase studies do allow patients to then get retreated for cycles on their next available dose. So if they are the first patient enrolled on your study and they're at the lowest dose, which again may not be an appropriate therapeutic dose, as you move up in dose escalation, a lot of the protocols I see are doing the patients a great advantage, are serviced by allowing them to, again, escalate with the rest of the patients. Those patients are not necessarily included in the scheme for DLTs because you're only looking at typically that first cycle of therapy. So that way you're not trying to do DLTs across a type of patient who maybe have treated at different dose levels several times, but at least that patient is being allowed to escalate to a next available dose to them so that they might get closer to a therapeutic dose for that patient. And you also get more safety data overall on your product as that patient increases. So keep that in mind for design too. All right, general limitations, phase 1 trials. Again, there's always going to be limitations of different designs and different phases. It's not a precise measurement, I would say, especially if you're using the older 3x3. It's why I think some of the newer designs are more helpful to get to your recommended phase 2 dose, your max tolerated dose. Again, you may have a safe product where you don't have a high amount of toxicity. So you're really going to need to look at biological activity. So again, there's going to be limitations into it. Other limitation, it's fairly slow to enroll these patients. I've seen the agency with a high risk new product really want to enroll 1 patient at a time in their first cohort, as opposed to allowing them to enroll 3 immediately, just to get some first in human safety with a new molecular entity. So kind of keep that in mind. The FDA may have some feedback on there, but that may slow it down. Again, these patients are going to be slow. There's a ton of competition. Again, it's a very crowded market. It's another reason it might be slow. Just because you're at a major academic study, don't think that you're the only phase 1 center study at that center. So just things to keep in mind.
Can I ask a question here?
Go ahead.
So for this phase 1 trial, so since this is not based on the evaluation of the efficacy, do you use health people or patients? So for example, if some of the patients, it's advanced cancer or the advanced disease, it's in the urgent, they don't have maybe a lot of time to live. If you use these and maybe they are toxic, maybe they are not effective at all, you basically delay the treatment options, right? I mean, is it ethically a problem?
That's a good question, actually, in terms of healthy patients versus cancer patients, especially as you get into therapies that you feel may have no toxicity profile that you would see. So it might be more optimal to study it in a healthy, normal population. So as long as you can paint the scientific rationale for your product and why it would be appropriate to do it in a healthy, normal population, it's actually a way to speed your way through that phase 1 study as well from a time perspective. But it also may limit the amount of patients that get a suboptimal dose. So from an ethics perspective, it's definitely a discussion that I think should occur more as we get into different therapies that don't have the same side effect profile that, say, chemotherapies have had, right? When they were first coming out, you were basically trying to kill the cancer before you killed the patient. You wouldn't give that healthy normal, but that's not the case with these therapies today. I have seen a client do that. They did take theirs into a healthy normal population, and they justified the fact with their specific product type that they had a very, very low risk of any sort of side effect profile. And so they went in and did a healthy normal volunteer study where they were allowed to do a what's called kind of a SAD MAD design, a single ascending dose, multiple ascending dose design, and we're able to do that fairly quickly. And now they're going back into targeted patients.
But on the other hand, if you use normal people, we know that many times that the patients and normal people, they have different tolerance, they have different microenvironment, they have different response to treatment, right? I mean, if it's safe in normal people, maybe it's not safe enough in the patient.
Yeah, you really have to evaluate the rationale to go into the different populations into a healthy normal population, especially if your plan for the product, especially as a therapy that you see would be specifically targeted into a cancer population. The profile of those patients is going to be different, as you said. So you really have to show why it makes sense to do it in healthy normal population. I've only seen it a couple of times, and it was a product that was, again, a very specific product type that essentially had no profile for toxicity within healthy patients. It did actually end up filing 2 different INDs, where one they were going into a cancer and one they were going into non-cancer indications as well. So that particular product had multiple uses. So they end up going into healthy normal first with that kind of other IND.
Okay, all right, thank you.
All right, let's see, the last few minutes, I know we're kind of getting close on time, so I'll just kind of go through these a little quickly because I'll show a few things on endpoints. But as you get into your phase 2, phase 3 trials, again, now you're really looking at efficacy and you're looking at endpoints. Here, you're also collecting that additional safety data. So don't forget that your other goal is always safety with your product. You're going to now be testing that typical dose in a phase 2 trial. You can see endpoints here. There's a lot of different types of endpoints, again, depending on a different cancer type and what you might be doing. So talking with the agency on design, talking with KOLs on the endpoints, always important thing to do. Typically, these are going to be a little bit larger. You're going to provide some sort of proof of concept in there to understand, are you getting appropriate efficacy, not just some sign. The design I'll throw up here as an example, because I still see it a lot in use today, is the Simon 2-stage design and basic statistical checkpoint. When you design it from the beginning, allows you to enroll so many patients, see if there's a response, and then decide if you need to enroll additional patients, and then see if there's a response. So basically a 2-stage look at how patients are responding. So I see this as a very common design still today. I've seen this even not just in a standalone phase 2, I've seen this in phase 1/2s where you enroll initially, and then you carry your selected dose right into your phase 2 into a 2-stage design. So a very efficient use of time and protocol, and very efficient use of patients in your study. So I still think that's why it's in use today. It allows you a quick look at that data to know if it's a go or no go onto enrolling more patients. So threw that one up here. And again, there's additional designs, but this is a simple one still in use today. I think this came out early 90s, can't remember, late 80s, early 90s. All right, other designs, you may take 2 doses, again, into your phase 2. You may randomize to high and low dose, especially if you're one of those products that doesn't have a huge safety profile where you end up with 1 MTD, maximum tolerated dose. You may end up kind of getting to a point where you're selecting a biological dose, and you may want to evaluate a low versus high dose. So that's a common design I see these days as well. Again, there's multiple designs. Randomized discontinuation design allows patients that are end up putting on to placebo to go back and switch to the active agent. That's fantastic for patients. Also gives you more data with your product. All right, so as you move from phase 2 to phase 3, you're going to look for confirmation of efficacy at this point in your design. So as we kind of talked about earlier, the surrogate endpoints versus the traditional endpoints. Kind of pull up the next little slide here. It's probably hard to see a little bit on the screen, but again, there's a link and I'll give you these slides, but this comes out of the guidance here that kind of shows you some of the traditional clinical endpoints and then some of the other surrogate endpoints for that. And then it also says surrogate endpoints for that accelerated approval. So There are other products that have already gone through that accelerated approval, and you can kind of go look up what they were approved specifically on for that surrogate endpoint. And then you can also look whether that surrogate endpoint actually has met or not met the clinical endpoint, because you actually have seen some products that were approved for an accelerated approval, and then they didn't actually meet the clinical endpoint that they had agreed upon with the agency. So they ended up getting that approval taken away, or they had to modify their labeling one way or the other. So this is a snapshot as you're kind of thinking about where you're going clinically. Keep in mind those types of endpoints that you're going to need to collect in your essentially your phase 3 trial. If you have enough funding, it's always a good idea. If it's not too much, you can kind of collect those also in your phase 2, or be continually looking at them in your phase 2 for survival as you start planning your phase 3, so you can get some early looks. Are you really meeting what you expect from a survival endpoint, if that's going to be your primary clinical endpoint, or and how is that looking against your surrogates? Again, this is a good discussion to have with your agency as you move into that that planning for your phase 3, so that end of kind of phase 2 meeting standpoint. All right, I won't go through all of this, but again, included it. This again from the guidance. There's pros and cons essentially of different endpoints. So advantages, disadvantages. So this is good, again, information as you're getting into later stage and evaluating. And then emerging endpoints. So I can't leave without kind of mentioning, again, there are other endpoints. So don't think that the traditional and these traditional surrogate endpoints are the only ones. But just keep in mind, there are other ones that have been used recently for approval. And so kind of know what those are if you're going in those cancer types. So start understanding what types of endpoints have been used for your particular area that you're going into. There's again another guidance link for that. But I see minimal residual disease often as a secondary endpoint, collecting that along the way in your phase 2. So understanding, is that going to be additional data? So you may collect some additional data in your phase 2 on different endpoints to start evaluating, is that going to be an appropriate surrogate for you? And if you have really strong data of that surrogate as you move from your phase 2 to phase 3, you might be able to use that data in discussions of a new emerging endpoint in discussions with the agency. All right, I know we kind of went through all the drug ones, so I kind of leave this one with the diagnostics as well. So again, just kind of different studies that you have from a diagnostic standpoint. You have to understand where you fit as a diagnostic, your non-significant risk device, significant risk. Again, typically it's significant risk here in the cancer space, because if you're a diagnostic, you're going to be using that diagnostic to direct therapy based on whether you've diagnosed them or not. So typically fall in that significant risk category, which means you're going to have to file a full IDE application, which again, similar to that IND application, but full IDE application requirements go into it. And instead of phases, you're going to use terms such as like your pilot, your proof of concept study. Those are your first studies. You may be gathering data in those initial studies, maybe establishing a training data set, or you may be establishing just proof of concept. And then you'll go into your pivotal study, which can be a clinical validation study for that product. And typically you're going to compare that against whatever that gold standard is for diagnosis. In most cases, solid tumors, it's biopsy results, whether they have confirmed disease or not. So understanding that that's probably going to be your study design, is making sure you compare yours, validate your product against the gold standard. All right, I know we're here getting to time, so I've got only a last couple of slides that I can share with you guys as we follow up. I'd rather stop here. Make sure if there's any last questions, happy to chat. I can stay on and chat, or I can have follow up and chat as well with you guys, but only had a couple of last slides. These are more on trial operations, but happy to share those with you guys on this deck, and then wrap it up here.
Hi, Matt. I have a question. So you mentioned about the timing of clinical trial. And my question is related to, let's say if we have an orphan drug or the target is same, which is being pursued by big companies, but the disease is different. Then in that case also running a clinical trial would be challenging for a startup company, or because the target would be same being pursued by big companies, but the disease is different. So in that case also, the small company should wait until the trial is finished, or they can still pursue.
It's a good question. I think from a clinical standpoint, that actually may be a benefit, that you have another product that actually has data out in the world in another indication. So if there's no competition in the indication that you're going after, then really there wouldn't be any hesitation in my mind from a logistical standpoint to work in the indication you're looking at, because really you're looking for a competition of patients, and competition of those sites for those patients. And so if you don't have competition in the tumor type that you're seeking, then I think that actually would be again in your favor. Assuming again there's other data, or there's going to be other data. Clearly, if there's a safety risk or something that happens in that particular type of product, that might be a concern, and you might have to explain why your product's better than their product. But in terms of timing, I would say it's actually probably beneficial for you to keep moving ahead, because actually then they'll know, hey, there's going to be data in another indication with that type of product, maybe in parallel to what you're doing, but not competition of patients. So you can still go after that patient type.
Okay, thank you.
Any final questions? Well, thank you everyone.
I have one here. To be very quick. So you mentioned the phase 1 IND filing. So after 30 days, if you don't hear anything from FDA, you go ahead. But if there is some feedback, you send back your response. Do you have to wait another 30 days, or you have to wait until you get a reply from FDA?
Typically, the agency will respond back to you to confirm if what you've provided is sufficient, so that they can meet that 30-day window as well in addressing. So you shouldn't have to wait an additional 30 days after that for an FDA response. Once the FDA is communicating with you, they typically communicate back and forth. You can address the concerns, and you're both working towards the goal of 30 days at that point in time. You don't have to wait an additional 30 days, but you do have to wait till the agency does say that you've successfully answered that issue or that question.
Okay, yeah, okay, that's what I'm asking. Thank you.
Yeah.
All righty, we're at time. Thank you so much, Matt. Really appreciate your help and support in this great presentation. And thanks for Proxima. To everyone else, we will share the slides with you guys. We will post them once we get them from Matt, and we will see you at noon for the next session.
Thank you. Thank you, Matt.
All right.
Okay.
Thanks,
Thank you.
guys. Take care.